following analysis will present the findings of the investigation for assignment 2 that explores the classification of histological images using neural network models. The goal of this investigation was to evaluate the performance of these models in different classification tasks. The methodology employed in the investigation began with pre-processing and exploratory analysis of the data. To prevent overfitting, the unique identifiers for each row and patient groupings were removed from the dataset. Metrics were chosen to accurately measure the performance of the classifiers in identifying positive instances, balancing precision and recall, and assessing the overall performance of binary and multi-class classifiers. To ensure unbiased evaluation, the data was split into training, validation, and testing sets. Class imbalance in target label was addressed by employing class weights. Class weights assign greater importance to minority classes, ensuring all labels have equal weightage. Other approaches considered while developing the solution were transfer learning. This technique involves leveraging pre-trained models, typically training on large datasets and adapting them to the specific classification at hand. And the other one was advanced regularization techniques to address the overfitting. Techniques like dropout, L1, L2 regularization, and batch normalization were contemplated. These techniques introduce an additional constraints during the training process to prevent models from memorizing the training data too closely. In the first task of predicting cancerous cases, a baseline model was developed using a CNN architecture consisting of multiple input layers, one hidden layer connected to a neuron, and one last dense layer of a sigmoid function that outputs the probabilities for two classes, which are cancerous or non-cancerous. The final model achieved a test loss of 0.29 and accuracy of 0.9. The model demonstrated improved performance with a higher F1 score and AUC score of 0.89. Moving to the second task of predicting cell types, a more complex CNN architecture was used as the baseline, consisting of 3 convolutional layers with an increasing number of filters, followed by max pooling layers to downsample the feature maps. The last layer of the model is a dense layer with softmax activation function that outputs the probabilities of the input image belonging to each of the 4 cell type classes. Through hyperparameter tuning, data augmentation, and class weights, the model's performance improved significantly, with the test loss decreasing to 0.67 and accuracy increasing to 0.78. Limitations of our models: we have addressed label imbalances, improved baseline models through various techniques, and evaluated the performance using appropriate metrics. However, our models still exhibit overfitting and required further regularization and improvements. The model for predicting cell type, although achieving reasonable accuracy, falls short in precision, recall, and F1 scores. Our approach shows promise, especially considering the initial positive results obtained with a small dataset. With further refinement, more extensive training, and the adoption of advanced techniques, we believe that higher accuracies and lower overfitting can be achieved. For the first paper, the dataset used for evaluation consists of 100 H&E stained histology images cropped from different areas of 10 whole slide images, covering a range of tissue appearances, including normal and malignant regions, as well as artifacts. The paper discusses a novel approach for detecting and classifying nuclei in histopathology images. The proposed method utilizes a locality-sensitive deep learning technique with CNN models. The authors addressed the limitation of the standard CNN-based methods and introduced their approach, which incorporates distance-based calculations and a weighted ensemble for improving label accuracy. The paper is relevant to our approach as it discusses a novel approach for detecting and classifying nuclei in histopathology images, which aligns with our classification of histological images explored in our approach. However, our approach focuses on CNN-based methods, while Paper 1 suggests exploring alternative classifiers such as SVM and random forests. An alternative approach to this paper could be other techniques such as SVM and random forests, which can be explored as alternative classifiers for nuclear detection and classification in histopathology images. These methods have been widely used in the field of medical image analysis and offer different advantages such as interpretability and robustness to noisy sound data. The researchers introduced the concept of deep residual nets, which are not only easy to optimize, but also exhibit superior accuracy with increased depth. The experiments demonstrate that these extremely deep residual nets surpass previous networks in terms of performance, achieving remarkable results on the ImageNet dataset. Through rigorous training and utilizing techniques like batch normalization and weight initialization, they have successfully trained these networks from scratch. The researchers achieved a 3.57 top 5 error on the ImageNet testset, surpassing other approaches and securing the first place in a classification competition in 2015. The concept of deep residual nets is introduced in the paper and relates to our approach as it discusses the performance of deep neural networks. However, our approach primarily focuses on CNN models for histological image classification, while paper 2 discusses the advantages of deep residual nets in terms of accuracy and depth. An alternative approach for this paper could be inception models or architectures, which incorporate multiple filter sizes with a single layer, can be explored to improve the representation capacity of deep neural networks. These modules allow for capturing information at various scales and can potentially enhance the performance of the network. The CCDC-HNN technique produced in this paper aligns with the objective of our approach, which is the classification of histological images using deep neural networks. The paper introduces an approach for early and accurate detection of lung cancer using CT images, while our approach focuses on histological images. However, the suggestion of exploring other advanced 3D CNN architectures like UNet or VNet is relevant to our approach's aim of improving the performance of the proposed methodology. The analysis delves into the major contributions of the proposed method, including the use of a database of lung images for CT scan image extraction, the extraction of crucial features from the CT scan images using deep neural networks, the utilization of a 3D CNN to improve diagnosis accuracy, and the ability to differentiate between benign and malignant tumors. Nevertheless, the paper mentions that additional research is necessary to verify the findings of, and overcome possible challenges, including obtaining sufficient high-quality training data and improving the comprehensibility of the DL models. Alternative approaches for this paper could have been exploring other advanced 3D CNN architectures like UNet or VNet, which could have been advantageous. These architectures are specifically designed for medical image analysis tasks and can potentially improve the accuracy and performance of the proposed CCDC-HNN technique. For the fourth paper, the study proposes an automatic deep learning approach for disease prediction utilizing supervised deep learning methods with 4 algorithms with performance metrics used including accuracy, font score, precision recall, AUC, and V-score. The analysis provides a detailed discussion of the results obtained by each model. The VGG16 model achieved an average performance compared to the other models, while the VGG19 model showed the second best performance, indicating that a 90-layer CNN architecture was better suited for the osteosarcoma detection. The ResNet 101 model exhibited the highest accuracy and V-score, indicating its superior performance compared to other models, along with the shortest training time. This paper proposes an automatic deep learning approach for disease prediction, which is similar to our approach's objective of disease classification using supervised deep learning methods. The suggestion of designing custom architectures tailored to the specific problem and dataset is also relevant to our aim of improving the model's performance. Alternative approaches for this paper could have been to use custom designed architectures tailored to the specific problem and dataset. By designing architectures that take into account the unique characteristics of the disease being predicted, it is possible to improve the model's performance by effectively capturing relevant patterns and features. To extend the system to handle the specific situation of the detection and extraction of individual cells from the original high-resolution histopathology images, we can leverage image segmentation techniques, specifically instance segmentation. Instance segmentation involves not only detecting objects but also segmenting them at a pixel level, which would enable us to precisely extract individual cells from the high-resolution images. A machine learning technique that could be utilized to solve this problem and further extend our approach would be the Mask-RCNN. Mask-RCNN is a popular instance segmentation model that extends the faster RCNN architecture. It can detect and segment objects in an image, making it suitable for our task. By training the Mask-RCNN model with annotated data, we can teach it to accurately detect and segment individual cells in the higher resolution histopathology images. As for similarities, our approach in assignment 2 and the extended approach both acknowledge the issue of losing fine-scale information in larger resolution images of cancer cells. Both understand the importance of addressing this challenge to improve the accuracy of classification or detection tasks, despite being done through different means. As for differences, the extended approach specifically addresses the challenge of fine-scale information loss by suggesting the use of instance segmentation, particularly Mask-RCNN. It proposes training the model with annotated data to detect and segment individual cells at a pixel level in high resolution histopathology images, unlike our approach for Simon 2. This yields more precise detection of individual cells from high resolution images. Furthermore, another difference between both approaches is that the extended approach utilizes the MARCS RCNN model, which is more complex architecture specifically designed for instance segmentation tasks. The mask RCNN model is an extension of the faster RCNN architecture, which combines object detection and instance segmentation capabilities, which requires greater complexity than the CNN models used for the first assignment, which were only for classification tasks. The same metrics used in our approach in assignment 2 can be used to determine the performance and accuracy of the resulting model from the extension. Metrics such as accuracy score, precision score, F1 score, and area under the curve. Furthermore, the mean average precision score can be used to determine the efficacy and performance of the RCNN model in relation to its performance on object detection and instance segmentation.